We're going to move on now to the DOPPS in-center hemodialysis study research updates. And next slide, please. And for this, I'd just like to bring in Ron Pizzoni, who's, of course, the great friend and great colleague with the DOPPS program, been with the DOPPS since the very start. And folks, many folks on the line know Ron very well. So Ron will be moderating this particular session. Ron.
Hi, Bruce. Yeah, good morning, and good morning, everyone, or good afternoon, good evening, depending on where you're located. It's really great to have this autumn update meeting. And Bruce, I thought we were doing the PDOPPS session actually at 9:30 today. So I thought that we were going to do the DOPPS session earlier. So I don't know if all of our speakers then would be available at this time.
No, Ron. Sorry, Ron. Just to clarify, this is actually the DOPPS session first. And anyway, yeah. So we're fine. If Chris, if you go back to the slide deck, there we go. Next slide.
Okay. Thank you very much. I was a bit surprised at the slide there. Yeah. So as Bruce has mentioned, we're just about entering the 25th year of the DOPPS study, and currently we're in the 7th phase of DOPPS, which started in 2018 and will extend to 2021. And currently we have 17 countries participating in DOPPS 7 and about 420 sites, including 7 countries from Europe, Belgium, France, Germany, Italy, Spain, Sweden, and the UK, all 6 of the GCC countries, 3 major cities in China, to Beijing, Shanghai, and Guangzhou, and Japan, which has been part of the DOPPS for many, many years, as well as here in North America, study sites from Canada and the United States. So then if we should go on to the next slide, please. Yeah, so we have 4 different presentations today, and as you can see here, and with the first one being led by Dr. Angelo Caraboyas, who's a research scientist at Arbor Research. And Angelo is going to present a really interesting research project on really description of early use of etyl calcitide and MBD responses in the United States. So if we go to the next slide, and Angelo, thank you very much for presenting this today.
All right, thanks, Ron. So I'm going to be presenting on etyl calcitide utilization and talk a little bit about etyl calcitide dosing and MBD marker response in U.S. HD patients. Next slide. So CKD, MBD, and secondary hyperparathyroidism are common complications in hemodialysis patients. There have been several large observational studies that have shown the mortality risks for high PTH levels, particularly above 600. And despite this, the proportion of U.S. HD patients with a PTH level over 600 has doubled in the last 10 years, from about 11% to 23%. And so clinical trial data have demonstrated the efficacy for etyl calcitide for reducing PTH levels, but there's not a lot of data out there as far as real-world effectiveness of the drug. Next slide. So we have a few different objectives in this study. We wanted to provide really an overview of etyl calcitide utilization in U.S. dialysis patients. And so we started capturing data. Our first observed use was was in April of 2017. So from that time point forward, we wanted to analyze the time to discontinuation of etalcalcetide prescription. We want to know the characteristics of those initiators. We want to learn a little bit about dosing patterns, as well as trajectories of PTH and other key MBD markers following initiation of etalcalcetide. Next slide. So our U.S. DOPPS sample during this time period tended to be between about 150 to 180 dialysis facilities. And from this sample, we were able to identify about 2,500 new users of etalcalcetide. And so for these patients, we analyzed time to discontinuation using Kaplan-Meier methods, and then we analyzed the different MBD markers, PTH, calcium, and phosphorus. We analyzed both the changes in the mean levels of initiators, as well as the proportion of patients who were in the target range. And we did that using GEE models. Next slide. So this slide illustrates the proportion of patients prescribed etalcalcetide in U.S. DOPPS. And on the left side, you can see from the first initiation we observed in April of 2017 through August of 2019, the prescription prevalence increased to about 6% overall. On the right side, you can see when we stratified the data by dialysis organization size, there's quite a distinction there between the 20% prevalence in small independent dialysis organizations compared to only 4% in large dialysis organizations. Because the large dialysis organizations make up more of the population, you can see why the overall percent, 6%, is weighted much more towards that 4%. Next slide. So in this table one, we just wanted to show the characteristics of those patients who initiated etalcalcetide. And we did this overall and also in a couple different strata. We wanted to look at it by dialysis organization size again, because the usage was so different, and also by prior cinacalcet use. And that was if the patient had been prescribed cinacalcet at any point during the 3 months prior to etalcalcetide initiation. And you can see that highlighted in red there are the key differences. And so if you look at the median PTH level of a patient who was prescribed etalcalcetide, it was about 760 overall, but by dialysis organization, you can see that in the large dialysis organizations, PTH level was over 1,000 among etalcalcetide initiators, whereas in the smaller independent units, it was actually more closer to 700. And also by prior cinacalcet use, you can see for patients who had not been prescribed cinacalcet and initiated etalcalcetide, the PTH level was a bit lower, around 700, whereas in patients who had been on cinacalcet and then switched to etalcalcetide, their PTH level was much higher when they initiated the drug, about 850. Next slide. So we were interested in dosing patterns of etalcalcetide, and you can see from the figure on the far left side, when the patient was first prescribed etalcalcetide, almost all patients received a dose of either 7.5 milligrams per week or 15 milligrams per week. And you can see that after a few months, many patients started receiving much higher dose levels, and after about 12 months, it was about a third of patients ended up receiving a higher dose level than the 7.5 or 15 that was observed. at initiation. Next slide. So this graphic illustrates discontinuation of etacalcetide during the first 12 months. You can see after 3 months on the drug, only 10% of patients had discontinued etacalcetide by that point. After 6 months, 19% had discontinued, and after 12 months, 30% of patients had discontinued etacalcetide. Next slide. So this is I would say the primary finding here is the trajectory of PTH levels following etacalcetide initiation. You can see from the mean PTH level when patients initiated etacalcetide was about 950, and you can see that the mean levels slowly decreased through the 12 months, and after 12 months, the mean level had declined about 40%, down below 600 to 566. And on the right side here is the proportion of patients with a PTH level focused on between 150 and 600. And so for new users, there was 33% of those patients were what we call in target, between 150 and 600, whereas 2/3 had a level above 600. But after a few months, the proportion of patients in that target range steadily increased, and after 1 year, it increased all the way to from 33 to 64%, with only 26% of patients over 600. Next slide. So we broke down the proportion of patients who were in target. We wanted to take a closer look and stratify that data on whether the patient had received senecalcet in the prior 3 months on the left side, and on the right side, we stratified by the PTH level at etacalcetide initiation. And you can see that on the left, the percent in target was about 30% for patients who had a prior senecalcet prescription, but those users still managed to get into target range 63% of the time after 12 months. And similarly, even if the patient started with a PTH level over 600, so of course target achievement was 0% by definition, and even in those patients, the target achievement was 60% after 12 months. So overall, it was about, close to between 60 and 65%, and even among those with prior senecalcet use or who had started with a higher PTH, target achievement still ended up around 60 to 65%. Next slide. So we were not only interested in PTH levels, but also calcium levels, particularly if there was any concerns about hypocalcemia. And you can see that on the left side where we show mean serum calcium levels, there was certainly a very large immediate drop in calcium levels following initiation of etacalcetide, from 9.2 down to a valley there of about 8.8, and even after settling in there, it reached a mean of 8.9 after 12 months. But we also note that the risk of hypocalcemia, if we define as less than 7.5, remained actually very low in the range of 1 to 2% throughout follow-up. And so that large decrease in mean calcium tended to push those patients into that low-medium range of 7.5 to 8.3 there on the right side, but very rarely below 7.5. Next slide. And so, in addition to PTH and calcium, we wanted to also illustrate the trajectory of serum phosphorus levels during those 12 months following initiation of etalcalcetide. And you can see there was a moderate effect on phosphorus levels. The mean levels started at about 5.9, and there was a decrease across those first 2 or 3 months, and with mean levels ultimately settling at around 5.6, so a drop of 0.3. And if you look at the percent in target, which we had defined as 3.5 to 5.5, the percent in target tended to increase, and the percentage of patients with the phosphorus levels over 5.5 decreased from 55% to 45% over the 12 months. Next slide. So to summarize these findings, in the 12 months following etalcalcetide initiation, patients had large and sustained reductions in PTH levels, and that was even the case among those with high baseline PTH over 600, or those who had been prescribed senecalcet in the prior 3 months and switched to etalcalcetide. There were decreases in serum phosphorus and serum calcium levels, although ultimately the risk of hypocalcemia remained quite low. And so in combination, we believe that these results support the utility of etalcalcetide as an effective therapy to achieve the KDIGO guidelines for CKD-MVD markers in U.S. HD patients. That's all. Thank you.
Thank you very much, Angelo. A really excellent presentation. These are really impressive findings. These first results from the United States regarding etalcalcetide use and MVD control. I'm really struck, for instance, in patients who had just previously been treated with senecalcet, and among those patients then who were then given etalcalcetide, only 1/3 of them were in the target range, but after receiving etalcalcetide, then 2/3 of those patients then had a PTH less than 600, and it's really impressive. I was just wondering if others in the audience have any questions regarding Angelo's presentation.
Hi, Ron. Dr. Akazawa had a question about, he said, how about GI symptoms?
Yeah, I don't think we've looked at that with regards to this work at this time. It's an excellent question, Dr. Akazawa, and is something that will be really interesting for us to look at in terms of the patient-reported outcomes that we have. But at this point, we hadn't looked at that yet, but we'll certainly look forward to doing that. Yeah, thank you very much for that excellent question.
We had another question that asked whether the... Oh, go ahead.
Brian, yeah, please go ahead. Thank you.
Yeah, we had another question that asked whether etalcalcetide use differed across countries.
We are beginning to look at that, and Angela, would you like to respond with regards to this in looks of etalcalcetide across some of the other countries in DOPPS?
Yeah, I'm probably not the best one to answer this question. The current study was entirely based in the U.S., so I'm really not familiar with the data ex-U.S.
Okay. Yeah. We've looked a bit in Europe, and for data that we currently have from about 5 of the 7 European countries, and it differs greatly across the countries, being 5% or lower in a couple of countries, but then other countries, it's as high as 30-35%. So there's quite a variation that we're seeing. And in Japan, as I recall, there's considerable use in Japan as well of etalcalcetide. So I think on the order of 20 to 30%, as I recall. So there certainly has been substantial uptake and use of etalcalcetide in a number of the countries.
And then also in Japan, there is use of evocalcid, so a different actually oral calcimimetic as well. So a lot of interest. And I think as Dr. Akazawa questioned, a real issue will be to what extent tolerability exceeds that with cinacalcet, and therefore folks are able to stay on therapy longer, whether that's with etalcalcetide and or, for example, newer oral agents such as evocalcid. Yeah, thank you.
Thanks, Bruce. Yeah, that's an excellent point. Okay, if there aren't any other questions, then we will then go on to our next presentation. And this is an interesting investigation of management of diabetes in the GCC and outcomes. And this work, we have a poster here at ASN this year, and as well as for the prior presentation by Angelo as well. And so this presentation, this work is being led by Dr. Saeed El-Ghamdi from Saudi Arabia, and the analyses have been carried out by Brian Bieber, who you just heard on the phone a couple of minutes ago. So Dr. El-Ghamdi is one of our country investigators from Saudi Arabia, has been such a great colleague, investigator to work with, and so we greatly appreciate Dr. El-Ghamdi's presentation today. So Saeed, thank you so very much for giving us. Please go ahead.
Okay. Thank you, Ron, for the introduction, and I'm really pleased to be here. And this is actually part of our multiple work on DOPPS data in the region. As you know, that diabetes mellitus is a major public health problem in the world, and especially in the Gulf region, where it's really affecting many of the dialysis population or as a causes of end-stage renal disease. And everyone knows that type 2 diabetes is very prevalent in the Gulf countries, which is about 33%. And there is multiple studies has shown that from each country and from all region, show that from 25 to 33% of the population had diabetes mellitus type 2, especially. And this is really have been found in one of the studies significantly associated with the gross domestic product. Up to 40% diabetic will develop eventually diabetic nephropathy and end-stage renal disease, and nearly 40% of the incident end-stage dialysis patients had diabetes, and with which affects quality of life and the survival too. Next one. The high mortality of diabetes in patients has been thought to be due to altered hemostasis of glucose by uric toxin causing insulin resistance, and there's also lack of target in the glucose control. So we don't know exactly which is the best for our patient dialysis, because we know that about 25 to 30% of our patient population, they have what's called burned out diabetes, where they require no medication. The hemoglobin A1C is considered the gold standard test for diabetic control in diabetic end-stage disease, but this not has been shown to be the best method. It's essential for non-dialysis population, but for dialysis population, we are not really sure what's the best way to look at as a control, and what is the target we have to look at. Next one. Among patients with diabetes in GCC, we have two phases of our DOPPS studies, phase 5 and phase 6 from 2012 up to 2018. And we have objectives describing the prevalence of hemoglobin A1C measurement among the countries, and describe the distribution of hemoglobin A1C among patients with diabetes. And then we look at the relationship between those hemoglobin A1C and the mortality. Next one. We had total about 2,274 cohort of patients analyzed for disease from the 6 GCC countries: Bahrain, Kuwait, Oman, Qatar, Saudi Arabia, Iran, other Emirates. And in two different phases, which I'll show it in another slide. And diabetic stress was based on the cause of end-stage disease on patients' record. I know this is not 100% perfect, but that's what we had, where we never actually mentioned that diabetes is the cause of end-stage disease was accepted as such. The exposure to hemoglobin A1C was taken as the only baseline hemoglobin A1C within the last 4 months from entry into the study, and outcome was all-cause mortality. We're looking into all co-founders. And the model we have taken is Cox regression adjusted for demographics, morbidity, serum creatinine, and KT over V. Next slide, please. If you look at all the diabetes prevalence, it's quite different as a cause of end-stage disease. It's 45% in Saudi Arabia and as high as 74% in Bahrain and Kuwait. And if you look at it's almost about 60% of the dialysis population, we believe that the cause for the end-stage disease was diabetes. Next one, please. When you compare between diabetic and non-diabetic, you find that the diabetic, they have 39% catheter use compared to 28% of non-diabetic, and as expected also that from this that single pool KT over V was lower in diabetic compared to non-diabetic. While when you look at comorbidities, understandably that they are much more in diabetes population compared to non-diabetic population. When you look at the treatment on the right side of the slide, treatment of diabetes and end-stage disease, insulin was almost 40% of the patients they are on, while sulfonylurea less than 10%, and DPP-4 was 5%, and other medication are non-existing, which is really quite understandable if we know that 30% of the patients they are not on any medication. Next slide, please. We tried our best to follow the guidelines, but as everybody knows that guidelines is something and reality is another one. Let's move on to the next slide. Here, actually, distribution of hemoglobin A1C among diabetic hemodialysis patients, and you could see that significant proportion of the patients they are less than 6. So hemoglobin is very well controlled, and almost 25% of the patients they are very high hemoglobin A1C in some countries where the hemoglobin A1C more than 9, and the majority of the patients between 7 and 9. Next one, please. When you look at the mortality, the least mortality was found in hemoglobin A1C around 7, but whenever you drop your hemoglobin A1C less than 6 or exceed 8, you tend to exponentially increase your mortality, and what we called a moderate U-shaped curve, which you could see it in this slide. So the lowest mortality will be around 7, which is not different from what's been described in non-dialysis population. Next one, please. The results, the summary of the results showing that the overall 60% of our GCC DOPPS participants were diabetic in all countries, but still there is some variability, which is ranging from 45% in Saudi Arabia to almost 75% in Kuwait. Compared to non-diabetic patients with diabetes, they are usually older, and they have fewer patients, less dialysis vintage, 1.5 compared to 3.0 years. and they have, of course, higher BMI of 27.6 versus 24.9. Insulin was the predominant treatment, almost 40% of the patient, and much less sulfonylureas and DB4, less than 5%. Diabetes were associated with elevated mortality, which has been looked into different models, but the adjusted mortality hazard ratio was 1.69% after adjustment. The measurement of hemoglobin A1C within the 4 months prior to enrollment was quite variable too, where Bahrain will have zero measurement, and 36%, 1/3 in Saudi Arabia, and almost 95 or almost in Qatar. Among diabetes patients with hemoglobin A1C measurement, the median hemoglobin was 6.8, but with the mean of 25% of diabetic patients with hemoglobin more than A1C more than 8. So there was a quite country range different from 13 to 33% with high hemoglobin A1C. And the moderate T-shaped relationship was established of hemoglobin A1C with mortality after the adjustment for various co-founders. Next one, please. In conclusion, diabetes is highly prevalent in GCC hemodialysis population. Almost 60% is really quite different from the rest of the world. And the hemoglobin A1C measurements remain variable across GCC countries, from 45% to 75% in some countries. There is a mean of 25% of diabetic patients with hemoglobin A1C is quite high, more than 8, with quite variability among countries. If you look at hemoglobin A1C relationship with mortality, it appears similar to that what's seen in other DOPPS region, like in Ramirez publication, Diabetic Care 2012, and Hoshino and KI in 2017, showed that the lower risk at 7% of hemoglobin A1C and 6.7% in Japan. So I think probably the mortality, which we found that almost the best was around 7, is similar to what has been described before by Ramirez and Hoshino. We think that there is further investigation related to frequency of measurement of hemoglobin A1C and what's the optimal hemoglobin A1C target and control hemoglobin A1C via various treatments, especially with the advent of new agents like GLP-1 and others, is warranted. And and I think probably the country, especially GCC, has to look into this, if I can call it, pandemic of diabetes, and look at various measures to try to control it from primary care to secondary care and to tertiary care. Thank you very much.
Dr. Al-Ghamdi, Saeed, thank you very much for giving this excellent presentation. And you can see here in the GCC, there's such a high prevalence of diabetes, actually very similar to what we see here in the United States as well. And I guess it's a question not only for the GCC, but for all countries. With the impact of the new guidelines, the new KDIGO guidelines that just came out regarding diabetes management in CKD patients, and those both on dialysis and not on dialysis, and if folks think that this will have an impact or in terms of how diabetic patients are being managed or not. And I was just wondering, Saeed, in the GCC, do you feel that the new guidelines will, at least speaking for the GCC, and and interesting to hear from others in other countries, whether the new guidelines will help in providing more sort of unanimous approach to managing diabetes. I'm just curious what your thoughts might be from your own practice.
Yeah, thank you for the question. I believe that the guidelines really, I've read it all. It's really beautiful, well written. But of course, on the dialysis aspect is not that much there is consensus. But nevertheless, we have actually what we call around 7,000 patients in Saudi Arabia. They are on what they outsourcing programs. And in this program, they are mandated. They have to follow the guidelines. And right now they're measuring hemoglobin A1C not as before. They're almost 2 times per year of hemoglobin A1C, and they have certain guidelines has been distributed that the patient has to be followed by their primary nephrologist and not to resort to endocrinologist unless there is a complicated case. And I think probably by just following those guidelines, we see much better improvement in the outcome because you will see that there is more measurement of hemoglobin A1C, more follow-up, and better tight control of blood sugar to whatever we have thought that's probably the best, which is around hemoglobin A1C around 7. Thank you, Ron.
Thank you very much. Yeah, I really appreciate that. I'm looking at the time here, and I think we will then move on to our next presentation. And this
Ron, I was just going to.
Oh, thank you, Brian. Yeah, go ahead.
Yes, there weren't any questions, but I was going to mention briefly that on some of the slides you'll see some QR codes, and those are for one of our ASN abstracts. And if you follow that QR code, it'll take you to the submitted abstract if that's of interest to you.
Oh, thank you. Yeah, I was wondering what that was for. Yeah, thank you so much. That's really great. It's nice to have that. Okay, so I'd like to next introduce Dr. Zhou Li from who helps to lead our study in China and been such a great leader of the DOPPS study in China. And if Dr. Zhou today is going to present some results from the China DOPPS study. So, Zhou Li, thank you so much for presenting today at our Autumn Update meeting.
Okay, thank you for your introduction. So because it's the first time we present DOPPS China here in ASN during the DOPPS session in ASN, so I just want to talk about how DOPPS was managed in China and some achievement. So we have a background. So we know that the population of with ESRD who need ART treatment is increasing very fast. So last year, the number of new ESRD patients entered hemodialysis or peritoneal dialysis is about 80,000. So this and the total number is 800,000. So it's the biggest patient number around the world. But the dialysis patients on hemodialysis, the mortality is relatively lower compared with USRDS data. So we think that maybe some practice pattern difference among different countries between US and China may be different, which causes the better mortality in China, Beijing. So the first I want to present how DOPPS is managed in China. You know that all the DOPPS activity is managed in NBPA. This is Nephrology Plus Purification Innovation Alliance. So this organization, under this organization, we have managed the CRO, which controls the data management, and also we have a QLT to manage the 14 sites in Beijing, Shanghai, and Guangzhou, the 3 major cities in China. So the DOPPS China data can represent the patient management pattern in main cities in China. So the data flow from DOPPS link into Arbor Research data site, and after data clean, so the data return to NBPA. So NBPA, we have a data management, data analysis team who can do the data analysis. And the proposals come from our QOLs or from all study research sites. So this is the all flow management of the DOPPS activity in China. So the first, and then I want to present some achievements in DOPPS 4 and some data was under analyzing in DOPPS 5. So let's see DOPPS 4. this is our first our first publication in the year 2014, and it is about the dialysis frequency and compared with 2 weeks per week, 2 times a week with 3 times a week. The result is that we don't found any significant difference between 2 weeks to 2 times per week or 3 times per week in quality of life and in some laboratory parameters. Although the 2 times per week person has a relatively lower standardized KTV and MPCR. So this is the first publication. The second, we compare those who dialyze 2 times per week and 3 times per week to see if there are any mortality difference between these 2 groups of patients. The result is that we don't find any mortality difference among these 2 groups. And yes, we don't find any mortality difference. So, but I think that the patient may restrict their diet and daily activity to maintain internal equilibrium of the potassium and internal hemostasis. And the third publication is about the anemia management in China, so in Beijing, Shanghai, and Guangzhou. In this study, although this study is using the data of 2011 or 2013, so we see in the study, we see that in the analysis, we see that although China and Japan achieved a similar hemoglobin level, the average ESA dose used in China is twice that used in Japan. So we hypothesized that this may be caused the quality of dialysis. We checked with the Japanese society. They told us all the dialysis is recommended to be ultra pure by their society. So in China, not doing this. So after this hypothesis, we recommend that ultra pure dialysis can be used in China, but they may be money consuming, I think. So the last publication is about the CKD-MBD. This is also using DOPPS-4 data in the year 2011 and 2014. We see that in this study, in China, we see the patient with who have PTH level greater than 16, the percent of patient is relatively higher compared with other regions, including Japan, North America, and Europe. Also, patients with higher phosphate level is also higher in China compared with other DOPPS regions. So this is might be because of the shortage of drug. During last 3 or 4 years, a lot of new drugs use the CKD-MBD management marketed in China. I hope that in DOPPS-5 and DOPPS-7, the CKD management will be better compared with DOPPS-4. So this is CKD. So this one publication using DOPPS-4 data, who is led by Chinese KOLs. And in DOPPS-5, we also are doing some ongoing analysis. This is include multiple mortality risks. Among these results, this is comparable with other publications in the literature. You see that the patients with lower albumin, with lower blood platelets, with chronic heart failure, they all have increased risk of mortality. You know that in China, the prevalence of hepatitis B or C, the prevalence is about 4%. Among these patients, they also have a higher mortality compared with those without hepatitis B and C. And in this analysis, we also see that patients with larger pulse pressure have higher mortality. This is maybe because of the rigidity of the vascular wall. So this is analysis undergoing. So we will make a submission for publication. The second analysis is undergoing. Next, please. is about the blood flow rate and the mortality. So these have communicated with Albert to want some input for this further analysis, because we know that blood flow rate is usually related with KTOV. But after adjustment of the standardized KTV, we still found that the blood flow rate less than 215 have a relatively higher mortality. So I think that maybe caused by the better removal of middle molecule. Maybe this can be explained by this, but further work needs to be done to validate this hypothesis. And this functional next please, physical function and mortality. In this physical function, next slide please. Please, next slide. So in this analysis, we see that patients with a lower ability of physical function have a higher mortality. All-cause mortality and first hospitalization all happen those that have a limited level of activity. So this is under publication. We have finished the draft and is submitted for publication. So this is how DOPPS China is managed, and some published the DOPPS data using DOPPS4, who is led by Chinese KOLs, and also some analysis undergoing for DOPPS5. In summary, so I want to thanks to all DOPPS China study sites, and some in DOPPS4 we see that anemia and CKD management need to be improved. And also we found some results comparing the 2 and the 3 sessions per week, and we found a similar quality of life and similar mortality, but this need to be more work to be done to see why this result. And also we see that the mortality in China relative, we see that is about 8%, not represented here in the slide, is relatively lower than some other DOPPS regions, including the U.S. So we think maybe we have some better practice pattern that can be referenced by other DOPPS countries or other countries. So in DOPPS7, you know that the DOPPS7 is undergoing in China. We hope that there are some more results coming from China that can be referenced by other DOPPS countries. We also want to thank the domestic and the international companies that for their support for DOPPS activities in China. Thank you.
Thank you very much, Dr. Zhou, for giving such an excellent summary of the study, of the DOPPS study in China, and the results from these number of papers already over the years. And it's such a robust research program. This has been so excellent. We greatly look forward to many more projects in the very near future and coming years. So thank you very much. This is really excellent. Thank you to all the DOPPS China study sites and all the other investigators in China as well. We greatly appreciate the participation of China in DOPPS. So thank you so much.
Thank you.
You're very welcome. Yeah. So now we'll be moving on to the fourth presentation in this DOPPS session. And this is a very interesting presentation presented by Dr. Golestana, and who will describe the role and place in the association of race with outcomes for hemodialysis patients in the United States. From Albert Einstein. So Dr. Golestana, thank you so much for giving this presentation today.
Oh, thanks so much for having me, and thank you for that introduction. Hopefully you can hear me as I unmuted myself. And I also want to thank ARPA Research for their amazing collaboration. The title of my talk was the role of place in the association of outcomes in patients on maintenance hemodialysis in the U.S. And the first of the studies I'm going to talk about on the next slide was just actually published in print late last week with an accompanying editorial by Dr. Zamaka and Cruz, which was very flattering. And the title of that paper is Community Racial Composition Hospitalization Among Patients Receiving In-Center Hemodialysis. Next slide. By way of introduction, there are over 700,000 patients with end-stage kidney disease in North America. They comprise 1% of the Medicare population in the U.S. and account for 7% of expenditures. 40% of these expenditures are from hospitalizations. And the USRDS actually has identified Black race as being associated with a higher incidence of hospitalization. Next slide. So what piqued my interest in setting about to do this study was, I saw this map from the USRDS, and it's the hospitalization ED visit rates across the country, and the variability really was pretty shocking to me. And I thought, well, if there's so much variability, there must be opportunity, and why is there so much variability? Next. So the hypothesis for the first study was patients receiving in-center hemodialysis in dialysis facilities located in communities with the highest percentage of Black residents, number one, are hospitalized more frequently, and number two, have lower dialysis quality, higher comorbidities, and exhibit lower adherence to dialysis, perhaps explaining number one. So by way of methods, we linked actually the DOPPS phases 4 and 5 databases covering years 2010-2015 to the American Community Survey, which is provided by the Census Bureau. And so our exposure variable was percent Black in dialysis facilities zip codes from ACS, divided into tertiles, 1, 2, and 3. From there, we did bivariate analyses to look at differences in the DOPPS-derived variables below, which were facility-level variables, patient-level variables, and the ACS provided the community-level variables. And then for our modeling, we use negative binomial regression adjusted to duration of exposure and to facility clustering to first examine incident rate ratio of hospitalization in the base model and in a multivariately adjusted model that we'll explain in a few minutes. Next slide. So this is our study population. We start off with 4650. And after we removed facilities without follow-up data and their subjects, we ended up with 4, 5, 6, 7 patients over 127 zip codes, and the bulk of them were from phase 4 DOPPS. Next slide. So here are some of the results. Demographics, total population as mentioned is 4,567. 53% were white, 27% were Black, and 45% were female. And the median percent Black residents of all the facilities, zip codes rather, that were examined was 5.5%. And the various tertiles 1, 2, and 3 showed a median of 1% in tertile 1, 5.5% in tertile 2, and 34% in tertile 3, with the interquartile ranges represented. Next. So when we did the bivariate analyses, we saw that actually those in tertile 3, those in communities with a high percentage of Black residents, were younger. They also had most of the patients, or a higher percentage of patients dialyzing in those communities were Black. Fewer had AV fistulas. The median Charlson score, comorbidity scores, were lower in those patients dialyzed in tertile 3 versus tertile 1. And diabetes was less frequent in tertile 3 than tertile 1. There's no difference in URR or hemoglobin. Next slide. As far as facility level variables, a higher percent in tertile 3 were for-profit, and the facilities seem to have more patients in tertile 3 as compared to tertile 1. The bulk of the facilities, actually all the facilities in tertile 3, were in urban communities, and about 17% versus 11.8% lived under the poverty line, so were poorer. They also had a higher median dialysis minutes prescribed per week in tertile 3 compared to tertile 1. Next slide. So when we look at the unadjusted model at the very left side of this table, what we can see is as compared to tertile 1, the incident rate ratio of hospitalization was higher in tertile 2 and significantly higher in tertile 3. As we adjusted for variables, with model 1 adjusting for age and sex, model 2 with clinical comorbidity, model 3, and please keep in mind these models are progressive, so model 2 is on top of model 1 and so on and so forth. Model 3 was adjusting for facility level variables, including dialysis prescription, URR. Model 4 was adjusting for patient-level variables such as adherence and drug use. Model 5 was then adjusting for community-level variables such as poverty, education, and other community-level attributes. And Model 6, the last model, was adjusting for all the other variables in the previous models and individual race and ethnicity. And as you can see in the fullest model, Model 6, as with all the other models, there was a persistently significant association or persistently higher IRR in tertile 3 as compared to tertile 1, which was statistically significant. Next slide. When we did interaction testing, and these are the analyses, stratified analyses represented, the first on the left, or A, being tertile 2 versus tertile 1, and B being tertile 3 versus tertile 1 of community racial composition, we found no effect modification by age, sex, individual race, facility profit status, Charlson score, or facility census. Therefore, we concluded, next slide, the patients receiving hemodialysis in communities with the highest percentage of Black residents were, I don't see here, they were younger, they were healthier, a higher percentage were Black, and they had a higher incident rate ratio for hospitalization. So next slide. So what is it about place that may be contributing to these outcomes, we were thinking, after we found these results? Is it the healthcare system delivery? Is it something else? What we noticed when we did the analyses was that hypertension as a cause of kidney disease was more frequently used as a diagnosis in tertile 3% Black residents as compared to tertiles 2 and 1. And next slide. Okay. And next slide. And hypertension being a rather controversial cause of end-stage kidney disease made us think, well, let's look at this a little more closely. Why is hypertension being used more commonly? And by the way, we noticed that glomerular disease was being used less commonly as a diagnosis or etiology of end-stage kidney disease in Black communities versus non-Black communities. So we decided to focus on Black men only so as to reduce any bias introduced by race and gender. And that brings us to our next study that we just got notification will be published in KI Reports very soon. And this is a summary slide. The title of that manuscript is The Role of Place and Disparities Affecting Black Men Receiving Hemodialysis. We decided to focus on Black men so as to reduce the bias introduced by individual race or gender. So we used the same DOPPS cohort. We limited to Black men only, and we combined it as we did. We linked it, so to speak, by zip code to the ACS. And so we were looking at a primary analysis of Black men receiving maintenance dialysis, a total N of 702. And we looked at the association of facilities level zip code percent Black residents with number 1, hospitalization incident rate ratio in patients treating in those facilities, and number 2, mortality hazard ratio in patients treating in those facilities. We then progressively adjusted for individual characteristics such as comorbidities, adherence, provider characteristics, and community-level variables such as poverty, education, and internet access. And our findings are summarized to the right of this slide. We found that total hospitalizations, we had 1328 events. The incident rate ratio for hospitalization was 1.09 per 10% increase in Black residents of zip code. And so this was statistically significant. And total mortality, a total of 97 events, hazard ratio showed increased hazard ratio 1.29 per 10% increase in Black residents per zip code. So therefore, we concluded that among Black men, if you can go back to the last slide, we're almost done here. Among Black men, receiving hemodialysis in communities with a greater proportion of Black residents was associated with increased hospitalizations and mortality. And finally, as yet identified community variables do play an explanatory role in the association between dialysis patients' race and their outcomes. Quality of local healthcare delivery may be a contributing factor. And that concludes my talk. I would like to thank all my collaborators, by the way.
Dr. Goldestana, thank you so much. This is such a interesting and great presentation. These results are so interesting, and I was just curious as to where do you think you'll be going from here? Which is indicated in your conclusion slide, but some of the kinds of things that you might be looking at then as you go from here.
I am trying to get my hands on the AHRQ databases to look at, perceived access to care by community, census tract, zip code, and actual access to care by some of the databases in the AHRQ. And I'd like to link them to the CRIC cohort and look at CGD progression to ESRD. To see if there's, what is the kind of explanatory association there. Thanks for the question.
Oh, great. Yeah, and these are such large effects that you're seeing here. So, well, thank you so much. It's such an excellent work that you and your colleagues are doing, and it's been wonderful to have the collaboration with the DOPPS program, and we're so happy to be able to contribute through the data that we have available. I was just wondering if anyone else has any other comments, or Bruce, if you have any comments of any sort.
Well, I would just note, thank you again, Dr. Goldestana. And this is from our standpoint, a wonderful example of data sharing with an external, very talented group of investigators. And of course, this has led to really productive work with publications, as you note, in AJKD and now in KI reports within short order. So this is just great. I mean, and we love to see use of these data for these purposes. So thank you once again. I think in the interest of time, we'll move on now to the section on peritoneal dialysis. And actually, we're doing great in terms of time. Actually, just about 5-6 minutes behind, which for us is actually quite good. But we'll keep pushing forward very, very efficiently. And I will note, actually, Jeff Pearl is listed as the moderator for PDOPS. Jeff actually has clinic this morning, Monday morning. But in his absence, I would like to anyway give a warm thanks to Jeff for all of his work with leadership of the PDOPS study. It's just tremendous. And as folks in the community know, Jeff is so efficient and does so much and is really a wonderful spokesperson with respect to the most recent in developments in the PD space and where the modality itself is headed in the future, et cetera. So just did want to give a warm thanks to Jeff. And as Jeff is not available, Ron Pizzoni will also